Okay, so we're going to go ahead and start describing photosynthesis, and the very first step are the light-dependent reactions. And full disclosure, just so you guys know, I don't know if it makes you feel better or not, but this is my third attempt at making this video because I've just tried 2 times before this and I messed it up. So let's see if I can get it right this time. So the purpose of the light-dependent reactions is to generate energy. When we talk about the light-independent reactions, that's where we're actually building sugar. And building sugar is going to cost us energy. Well, in order to build something that's going to cost us energy, we have to have energy to burn. And so the light-dependent reactions aren't really about sugar yet. They're about making energy so that we'll have it available to do the next set of reactions, which is where we actually produce the sugar. So in the light-dependent reactions, we've got 2 big blocks of proteins. So in respiration, we had the protein complexes. Here we've got these photosystems. And they're the same idea. They're big old blocks of hundreds and thousands of proteins that are all embedded inside of a membrane. And so I've got one big block of proteins, another big block of proteins, and then a membrane that you could find both of them in. The one that's going to come first is called photosystem 2, and the one that comes second is called photosystem 1. And so the names are kind of goofy. This one's going to come first, this one's going to come second. So you might be like, well, then shouldn't this be number 1? But the reason is that photosystem 1 was discovered first. Then they discovered photosystem 2. Then they realized that, oh, well, 2 actually comes before 1, not after it. But by this point, they already published a bunch of papers calling this one number 1 and this one number 2. So they're like, whatever, we'll just keep them, the names, the way they are, and they'll just be the named in the order that they were discovered, rather than the order in which they occur. And so that's why number 2 comes first. And so in between photosystem 2 and photosystem 1, we have something else, and it is a group of proteins. And so we've got this big chain of proteins, and they're embedded in the membrane just like the photosystems. And if you think back to respiration, we also had a chain of proteins that was embedded in a membrane. And the reason that they were embedded in a membrane rather than just kind of floating around is because they were carrying electrons. And so because they're carrying electrons, which is potentially dangerous cargo, they've got to make sure that they're inside of a membrane. And the same thing's happening here. This is a chain of proteins that is carrying electrons. And so it is called the cytochrome complex, but the cytochrome complex is really just the same thing as the electron transport chain, which we saw in respiration. It's like the same proteins. It's totally the same thing. So now we can talk about what's actually going to happen. So PS2 is this group of a whole bunch of proteins, and the very first protein that's going to be named within PS2 is a very special one that is called P680. And P680 is going to do a really special reaction where it is going to take a molecule of water, it's going to split it, and it's going to split it into oxygen, so obviously it's more than one water molecule, and a hydrogen proton. And if you think about what's going on, you got a hydrogen proton, so hydrogen, just normally as an atom, has 1 proton and 1 electron. But if it has a positive charge, that means it's actually missing an electron, and so it's just a proton. And so P680 is able to do an atomic or nuclear reaction in which it's going to split the particles that are within these atoms to extract an electron. The electrons are actually going to come from the oxygen rather than the hydrogen. Now, this is kind of a small detail, but the idea is that P680 is a very, very special protein that is one of the only ones that we know of that is able to do this atomic kind of reaction where it's going to obtain an electron from these donor molecules, which in this case is water. So now that it has that electron, it's going to energize that electron, and the energy to energize it is going to come in the form of photons from sunlight. And so up here at the top of PS2 is where you will find the chlorophyll pigments. And those chlorophyll pigments, they are absorbing the incoming photons from the sunlight. Once they absorb those incoming photons and they have that energy, they're going to give it to this electron. And when they give it to that electron, the electron's going to raise to a higher energy state. And so inside of this picture If you are vertically high in this picture, that means you have high energy. And if you're down here in the lower part, you have low energy. So that's why we have this arrow to indicate that it is now high energy. So this high energy electron, we could do lots of things with it. We need high energy electrons that need to be inside of our sugar, but we can also use high energy electrons to do work, just like you saw in respiration. And so first, we're going to put this high energy electron to work. And this high energy electron, it's going to go down the cytochrome complex from one protein to the next. And we're going to use it just like we did in respiration. And if you go back to thinking about the electron transport chain in respiration, it pumped protons. So we're going to do the exact same thing. We're going to pump protons across this membrane. And if we're having to use energy to pump protons, which is active transport, that should tell you something. It should tell you what the charge is of this side of the membrane. And it is positive. The reason that we know it's positive is because it is costing us energy to pump the proton over here. If this was negative, then the positive would want to just come over. But because we know that this side is positive, that's why it's going to cost us energy to move another positive over here, is because it's against the concentration gradient. So anyways, we absorb energy at photosystem 2. We give it to the electron, which was gathered from water at P680. That energetic electron then goes down the cytochrome complex to pump a proton, and it's going to arrive at PS1. At PS1, we're going to repeat what we did before. We still got sunlight. Sunlight's going to give us some photons. So that photon of energy is going to come in, and it is going to raise this electron to a higher energy level. So now we have another energetic electron. At this point, we could do the same thing. We could send it down another electron transport chain. But we really don't need to, because we've already done that. We've already pumped a proton. So there kind of isn't any reason to go through that whole step. So instead, we're going to carry this electron and use it inside of the glucose that we're eventually going to make. And so this electron is going to get carried away so that we can use it for a later step. And the molecules that carry electrons are NADH and FADH inside of respiration. So we're going to use the same kind of molecule, except in photosynthesis, instead of NADH, it's NADPH. So this electron is going to be combined with the molecule of NADP and a hydrogen proton to give us NADPH. And NADPH is carrying that high energy electron. So you might say, okay, well, what do we care? Who cares? Why do we do this step with the protons? And it's because we're going to use those protons the same way we did in respiration, which is that if you're looking at the parts of the chloroplast has got two membranes on the outside. Okay, and these membranes have really boring names. This is just the outer membrane. This is the, you guessed it, inner membrane. The space on the inside, if it was a mitochondria, it was the matrix. In a chloroplast, this is called the stroma. And then there's one more membrane on the inside, and this membrane, it's called the thylakoid. And it's one continuous membrane that has a very intricate folds. And so the way we're going to do it is it comes out, over, and so we're going to call this membrane the thylakoid membrane. And this thylakoid membrane, I drew little squiggles, but really this is like little hockey pucks. It's like, imagine a stack of pancakes or a stack of hockey pucks all on top of one another. And so they're folded into these little packets, which means that inside of that little packet is a little empty space, a lumen. And this picture that we just drew of the different light-dependent reactions, this membrane right here, this is the thylakoid membrane. So this membrane that we drew right here is this membrane. And this area that is full of protons is the area inside of one of these little packets. So if we were to zoom in and see that little packet that's right here, the inside of those packets would be positively charged, because the inside of those little packets is where we're pumping all of these protons. The other side of the membrane, this out here, this is the stroma. This area out here, that is the stroma, which is in this picture. The stroma has a negative charge. So since we've got all these packets full of a positive charge, an outer area that has a negative charge, we can use an ATP synthase, just like we did in respiration, to make ATP. And so there are ATP synthases inside of this membrane, just like there are photosystems, and they are producing ATP when the protons go out of these membranes back into the stroma. So the products of the light reactions are NADPH and ATP, which is going to be produced from this proton gradient that we're creating across the thylakoid membrane.